Ready? We're back, and I want to just pick it up from the slide that we left off on. So our focus now is going from talking about the biochemistry and the characterization of the cells that are responsible for detecting the light stimulus and talking more about how the information is actually processed. I hinted at this in talking about the receptive fields earlier, and now what we're looking at is that we're going to try to delve into this concept a little bit more detail. We'll revisit this again on Tuesday when we meet again to ensure that the concepts are well grounded, but it's something that we can begin at this point. So, alright, so what's the deal? So in the outer plexiform layer, where we have the connections between the photoreceptors down here and the bipolar cells here, there's a lot of communication and shaping that occurs with the between the different cell types. And so, as we know, photoreceptors are releasing their neurotransmitter when they're actually depolarized. But what we also have is that we have these bipolar cells, which we've been introduced to here. And these have receptive fields of multiple different types of photoreceptors feeding into them if they're in the peripheral retina, and much more focused if they're in the central retina. So this kind of depiction here, we've seen this kind of drawing at the very beginning. And then if we look at one that has a little bit more of a schematic, we can see this depiction of the photoreceptors being shown here, and then the horizontal cell being shown here and the bipolar cell here. So the orientation of this is flipped from what we have here, but we can see photoreceptor, bipolar cell, ganglion cell would be over here. So this direct pathway that we've talked about before is illustrated in this picture here, where light's shining into a center or a focused area of this particular receptive field. And then what we're looking at is that it's actually causing depolarization of this bipolar cell. So here's the thing that's happening. If you move the light over, and so instead of hitting it in the center and activating this bipolar cell as we kind of described in the direct pathway right here, if instead of striking light here, instead we strike it over here in this outer area, this information gets carried to the bipolar cell. But the horizontal cell that's present here helps to shape it so that that information ends up having a different kind of influence on the bipolar cell's activity. And instead of causing it to become instead of it becoming depolarized in the presence of this of the light, the bipolar cell becomes hyperpolarized. So let's kind of go through that again and make sure we're kind of seeing each of these different parts here. So we'll start off, here's our bipolar cell, here's our photoreceptor, and there's clusters of these photoreceptors. This is sort of a cutaway of it, and so we're seeing sort of a semicircular thing that ultimately, if we were to look at the entire circle here, it would appear as this kind of appearance here. The receptive field, in other words, of this bipolar cell includes these clusters of cells that are depicted here. So now the next question then is, well, if light comes in and shines on one part of it here or one part of it here, how does this bipolar cell respond? Well, as we know, light, when you shine it onto a photoreceptor, makes it become hyperpolarized. And that's true no matter where you shine it, whether you shine it in this blue area or whether you shine it in the red area, as is shown in this image here. But how the bipolar cell responds to that is different. If you shine the light into this center area here, then the bipolar cell becomes depolarized. If you shine the light onto this surrounding area here, then the bipolar cell becomes hyperpolarized. And depolarization for a bipolar cell is sort of like what you think of for depolarization of a regular cell. It's going to cause us exocytosis and all of those things that we typically think about. Okay, so these represent the formation or the shape of these receptive fields for bipolar cells. And here's a zoom in of those particular components that you can go and look through at your leisure. If we go ahead and then consider this schematic here, this helps us to be able to understand why do bipolar cells turn on and off, or what are the different types of orientations that these bipolar cells can end up having. And so what I wanted to do was to show you guys this kind of depiction here. So in this case, we're shining light onto the center of the receptive field of a bipolar cell. And what's interesting is that there are bipolar cells that can pick up on that center area that are on center, and there are also bipolar cells that represent the same area, but they're what are known as off center. And the reason they're described as being on center or off center is we're trying to characterize how they behave when you shine light onto the center of their receptive field. So to recap then, what we're talking about here is we have a cluster of photoreceptors up along here. We have light being shown on this, and for this bipolar cell, it happens to have a receptive field of these three photoreceptors. For this bipolar cell, it also happens to have these three photoreceptors. When we shine light onto the center of those three photoreceptors, it hyperpolarizes this photoreceptor. Great. That's something that we've learned about before. But now what happens is that hyperpolarization, which makes it so that there's less neurotransmitter being produced, excites this bipolar cell. So shining light into the center here creates and makes this on-center bipolar cell become depolarized, and then that in turn activates a ganglion cell and passes it onwards. So this is the trickiest part of the receptive field story, and we just want to make sure that's really clean and clear. Let's consider the off-center as another case in point. In this case, this off-center bipolar cell has three different receptors present. When we're shining light onto the center of its receptive field here, that hyperpolarizes that photoreceptor. So we have less of this neurotransmitter being released. So what that means is that this bipolar cell actually gets hyperpolarized. So bipolar cells can differ in their response to the same stimulus here. And the change, or the reason for that, is because of how these cells are actually structured. Before going into the next slide over here, let's just take a look at this and explain why this is the case. It has to do with the receptors that are present here and here. So this image helps us to be able to understand the on and off bipolar cell. And we're going to look over at this point right here to help illustrate it. By shining light onto a cone here, that causes what we described before, the hyperpolarization. So what typically is released as a neurotransmitter is glutamate from the cones there. And what ends up happening is that on an on bipolar cell, this is an inhibitory glutamate receptor. So what I want you to do, and what we'll go through when we meet again, is just go through and think about what that means here. You stimulate this, or you shine light onto this cone here. What happens to neurotransmitter release? Does it go up or go down? What happens to the membrane potential in the cone? Does it go up or does it go down? And then think about, okay, now that you have less or more neurotransmitter being released, what happens to the impact that has on this cell here, the inhibitory glutamate receptor? That in turn will cause this cell to change what it releases, which happens to also be glutamate as a neurotransmitter. Okay, so give yourselves a couple of moments to think about what that is, and I'll go through it now. Okay, so maybe you took a second to do that. These in-class exercises are hard to do online, but here's the deal. right? You're shining light here, so now you have your cone. Your cone ends up getting hyperpolarized, right? Because dark current causes this constant release of glutamate from the cones here. So now you're shining light on it, so you're getting less dark current, and now you're getting less glutamate released. And so you have less glutamate being released here, means that it can't inhibit this bipolar cell as much. And so now you have less of an inhibitory signal, so this cell will fire more. It's complicated, isn't it? All right, well, let's go through an off bipolar cell. In this case, if you get this, the only thing you have to change in terms of understanding that mechanism is that instead of having an inhibitory glutamate receptor, you have an excitatory glutamate receptor. Light comes in, shines on the cones. The cones, as we've learned before, become hyperpolarized, so they don't release as much glutamate. And because glutamate is an excitatory neurotransmitter for this particular cell, then having less glutamate around means that the cell is not going to fire as much. And so that's how those two cells are able to respond to the same stimulus differently. It's because they have a receptor that responds oppositely to the stimulus being presented. And so when they call it an on bipolar cell, this means that it's like an on center, if you will, bipolar cell. And this would be an off center bipolar cell. So let's go back and take a look at this. In this depiction then, we could actually help to describe that, okay, with an on center bipolar cell, these guys have inhibitory glutamate receptors on it, so that when there's less glutamate being released from this hyperpolarized cell, then you get more firing, and then that's passed on to the ganglion cell. The opposite here. Fine, great. What about if you move that light over to the surround? What happens then? In that case, what we find is that if we shine light here, here's the receptors that are being activated. They get hyperpolarized. The horizontal cells, I was describing that plexiform layer that's influencing the action of the bipolar cells and everything. So the horizontal cells help to shape the function of this and the release of neurotransmitter from the center photoreceptor here. And so these horizontal cells end up being able to translate this change in neurotransmitter release, there's less of it here, and ultimately making it so that this ends up having more depolarization when there's less light around. And so what you end up having is that you end up causing more depolarization here. So even though you aren't necessarily directly stimulating this, you've moved it over. The dark current, in addition to the communication of the horizontal cells to this particular type of photoreceptor here, helps to shape the response of those neurotransmitters. And so now what you have is that your on-center bipolar cell is going to be exposed to more glutamate, and that has inhibitory neurotransmitter or inhibitory receptors on it, so it's going to shut down. And so it's on-center. And the other way of describing it is that it's off-surround. So the surrounding is when you shine light on the surrounding receptive field, it's not going to fire as much, it's going to fire less. The converse is true here. Off-center here means that when you're shining light onto the surround, not on the center itself, then this guy's going to fire more than it did when you were shining light onto the center here. So that kind of switch between on-center, off-center, and on-surround, off-surround helps to characterize one of the basic kinds of structures that we see with how sensory systems are actually constructed. The really kind of cool element of this is that this helps to sharpen and the contrast between where a light is, whether it's shining here or whether it's shining here, is accentuated by this effect of the horizontal cells here. And being able to see edges or differences and accentuating contrast is one of the key and important elements for sensory physiology. So what I'm going to do is I'm going to stop there. When we return on Tuesday, we'll pick up here. It'll give us a chance to kind of go back and make sure we've got these concepts locked in. And then we'll move into describing how these things work together in this image here. And then we'll start talking about some of these different types of outputs that we have, M and P cells, and then ultimately link it to how the LGN is passing information up to the cortex. All right, so thanks for your understanding for having today's class online, and I'll see you on Tuesday.